Hello friends. In last lecture we have started with the neuromuscular system. Now let's discuss about the electrical activity of the neuromuscular system and electromyography. First we will like to discuss about the motor unit. Skeletal muscles are excited by stimulation through nerve fibers. The external process of a nerve fiber forms a junction with the skeletal muscle membrane known as the neuromuscular junction. The nerve cells which form the junctions are known as motor neurons. A single motor unit consists of one motor neuron and all of the muscle fibers it innervates. The cell bodies of motor neurons are located within the brainstem or spinal cord. The axons of these neurons are myelinated and large in diameter. That means they are able to propagate action potential at high velocities. The region of the muscle membrane which lies directly under terminal portion of the axon is known as motor end plate. Once an alpha motor neuron is activated to produce an action potential, all of the fibers innervated by this neuron are activated and contract simultaneously. Groups of motor units often work together to coordinate the contractions of a single muscle. All of the motor units within a muscle are considered as motor pool. When the motor unit is activated, it produces a motor unit action potential which is known as MUAP. The action is repeated continuously for as long as the muscle is required to generate the force. Now let's see the muscle excitation and contraction coupling. The figure shows the sequence of events by which an action potential in the plasma membrane of the muscle fiber leads to force production. The terminal ends of the motor axon, that is known as axon terminal, contain membrane bound vesicles. Those are synaptic vesicles. These vesicles contain the chemical transmitter which is known as acetylcholine, and commonly we are calling it as AC. Both are capital and then small letter h. Now, when an action potential arrives at the neuromuscular junction, it depolarizes the nerve membrane and releases acetylcholine into the space separating the nerve and the muscle membrane, that space is known as synaptic cleft. The acetylcholine diffuses across synaptic cleft and combines with the receptor sites on the motor end plate, depolarizing the end plate, which results in muscle excitation. The inward excitation also activates calcium released from the sarcoplasmic reticulum. The calcium then binds to the thin filament and cross-bridge formation occurs, leading to muscle contraction. This further shortens the muscle and produces tension. Further, the motor end plate membranes also contain the enzyme acetylcholine esterase, which destroys acetylcholine. The molecules of this acetylcholine released from the motor neuron endings have a lifetime of only 5 milliseconds before they are destroyed by this enzyme. Once they are destroyed, the muscle membrane returns to its resting potential. Now let's look how the muscle acts. A cell is basically existing in its two states, polarized or depolarized. So we are calling it as bistable device, and its immediate potential levels are not stable. Motor nerves are also cellular in nature, thus can exist in polarized or depolarized state, and will transmit only two potential levels to the motor end plates, causing a bistable on-off action of the muscle fibers. Normal muscular activity is characterized by the smoothness of movement, steadiness and precision. These characteristics are due to large number of motor units comprising one muscle. If a small muscle effort is required, only one motor unit will be called into action. As increasing muscular effort is required, many more motor units are called into action until the muscle provides maximum effort. An individual motor unit can result in only one level of muscle contraction. The number of times that this contraction occurs will effectively increase the power of the muscles. The number of muscles those are recruited depends upon the activity in which the body is involved. Thus, the smoothness of movement of muscle is controlled both by the number of motor units activated and by the rate at which these motor units are being activated. For example, in lifting a small weight such as a tiny pebble, fewer amount of muscles will be involved as compared to lifting a heavy weight, like for example 8 kg weight, where the muscles employed will be greater. In other words, we can say whenever it is required to generate greater force, the excitation from the central nervous system increases more motor units and the more motor units are activated. Further, let's discuss about human motor coordination system. We'll look one example in this. What happens when our hand suddenly touch any hot object? The sensory neurons carry information through sensory receptors, normally it is skin hair, and that information will be carried to the central nervous system. That can be either brain or spinal cord. Here it is spinal cord. Then spinal cord will respond through motor neurons, and the message from the motor neurons makes the muscles to contract, and thus our hand is pulled away immediately. Now let's see what we mean by electromyography or electromyogram. Electromyography is a technique for evaluating and recording the electrical activity that are produced by muscles. Myo is actually another word for muscle. When muscle contractions and relaxation occurred, the human brain will generate commands through neurons that are responsible for different movements of human skeletal system. These kinds of commands are carried through the nervous system, which will generate an electrical activity in the muscles. The electric potential that has been generating by the muscle is recorded, and this is called electromyography. The EMG signal is a measurement of electrical currents generated in muscles during its contraction. It is a result of the summation of all motor unit action potentials, that is, previously also we have talked about this MUAP, in the region near the electrodes. Now let's discuss about the different types of electrodes that are used to obtain an EMG signal. The bioelectrical activity inside the muscle of a human body is detected with the help of EMG electrodes. There are mainly two types of EMG electrodes, the surface or they are skin electrodes, and also other one is inserted electrodes. Further, inserted electrodes can be the needle electrodes and the fine wire electrodes. Needle electrodes are widely used in clinical procedures in neuromuscular elevations. The tip of the needle electrode is bare and used as a detection surface. You can also see in this figure. It contains an insulated wire in the cannula. The two main advantages of needle electrodes are that they are relatively small pickup area, which enables the electrode to detect individual MUAPs during relatively low force contractions. And also these electrodes may be conveniently repositioned within the muscle. Now next is fine wire electrodes. Wire electrodes are made from any small diameter, highly non-oxidizing stiff wire with insulation. Alloys of platinum, silver, nickel and chromium are typically used. Wire electrodes are extremely fine. They are easily implanted and withdrawn from skeletal muscles, and they are generally less painful than the needle electrodes, whose cannula remains inserted in the muscle throughout the duration of the test. Then next is surface EMG electrodes, and also we are calling it as SEMG electrodes. These electrodes provide a non-invasive technique for measurement and detection of EMG signals. These electrodes form a chemical equilibrium between the detecting surface and the skin of the body through electrolytic conduction, so that current can flow into the electrodes. These electrodes are very simple and easy to implement. Application of needle and fine wire electrodes requires strict medical supervision because they have to be inserted. Whereas if I talk about SEMG electrodes, they do not require any such formality. These electrodes have limitations. Since the electrodes are applied on the skin, they are generally used for superficial muscles only. And also, crosstalk from other muscles is a major problem. Their position must be kept stable with the skin, otherwise the signal will be distorted. Now, there are further two types of surface EMG electrodes. These are the gelled EMG electrodes and dry EMG electrodes. Gelled EMG electrodes are basically they are containing a gelled electrolytic substance as an interface between the skin and electrodes. Oxidation and reduction reactions take place at the metal electrode junction. Silver chloride is the most common composite for the metallic part of gelled electrodes. This layer allows current from the muscle to pass more freely across the junction between the electrolyte and the electrode. Special skin preparations and precautions such as hair removal, proper gel concentration, prevention of sweat, etc. are required for gel electrodes in order to acquire the best possible signal. Now next is the dry EMG electrodes. These electrodes do not require a gel interface between the skin and the detecting surface. These electrodes may contain more than one detecting surface. These electrodes are usually heavier, we can say greater than 20 gram as compared to gelled electrodes which are less than 1 gram. This increased inertial mass can cause problems for electrode fixation. Therefore, a material for stability of the electrode with the skin is required in case of dry electrodes. Now we have two different categories of surface EMG electrodes. They can be passive, they can be active. Passive EMG electrodes should be connected to an external amplification circuitry with the help of connecting wires for the proper acquisition of the EMG signal. These electrodes can be disposable or reusable. Gelled and dry electrodes both fall under passive surface EMG electrodes. Now, active EMG electrodes contain a pre-amplifier attachment for surface electrodes. The in-house high impedance amplifier in these electrodes transfers the pre-amplified signal to the rest of circuitry. Now further, let's discuss about electrode placement and signal acquisition. In this, first we will start with the skin preparation, where electrodes need to be placed or inserted. As the surface EMGs are the most commonly used because of non-invasive nature, for that proper skin preparation is required. In order to obtain a good quality EMG signal, the skin's impedance must be considerably reduced. For this purpose, the dead cells on the skin, for example hair, must be completely removed from the location where the EMG electrodes are to be placed. It is advisable to use an abrasive gel to reduce the dry layer of the skin. There should be no moisture on the skin. The skin should be cleaned with alcohol in order to eliminate any wetness or sweat on the skin. Next is how we are placing the electrodes. Basically, there are two detecting surfaces, or we can say EMG electrodes, that are placed on the skin in bipolar configuration. We will discuss there can be two different type of configuration, monopolar and bipolar. We will see in coming slides. The surface EMG electrode should be placed between the motor unit and the tendinous insertion of the muscle along the longitudinal middle line of the muscle. The distance between the center of the electrodes or detecting surfaces should be only 1 to 2 centimeters. The longitudinal axis of the electrodes, basically which passes through both detecting surfaces, should be parallel to the length of muscle fibers. You can see in this figure also. The EMG detecting surfaces should be placed in between the motor unit and the tendon insertion of the muscle. Detecting surfaces placed on the belly of the muscle has proved to be an acceptable location. Here the target muscle fiber density is the highest. When the electrodes are arranged in this way, the detecting surfaces intersect most of the same muscle fibers, and as a result an improved superimposed signal is absorbed. The electrodes should not be placed elsewhere. The electrode should neither be placed at or near the tendon nor at the edge of the muscle. The muscle fibers become thinner and smaller in number when they approach the tendon of the muscle, resulting in a weak EMG signal, providing the fact that electrode placement near the tendon is not feasible. So, always a proper electrode placement is necessary. Now, let's go through some general properties of EMG signal. The EMG signal's amplitude lies in between 1 to 10 millivolt, making it a considerably weak signal. The signal lies in the frequency range from 0 to 50 Hz and most dominant in between 50 to 150 Hz. The EMG signal is highly influenced by noise. Ambient noise can be basically caused by electromagnetic radiation sources, example radio transmission devices, fluorescent lights, and power line interference from electrical wires. These different sources we will study in the chapter based on sources of noise in these bioelectrical signals. Next is how we are placing the reference electrode. The signal from the EMG detecting surfaces is gathered with respect to a reference. An EMG reference electrode acts as a ground for this signal. It should be placed far from the EMG detecting surfaces on an electrically neutral tissue. EMG signal is acquired through differential amplification technique. The differential amplifier should have high input impedance and very low output impedance. That is mainly what we need for an amplifier. Differential amplification is achieved with the help of, we are using here, instrumentation amplifier for high input impedance. The instrumentation amplifiers carries out differential amplification by subtracting two voltages, because we are keeping two voltages as input, so it will subtract and amplify. We will show in the figure in the next slide. This way the noise signal which is common to this V1 and V2, in that case that will be eliminated. Now this is a diagram showing instrumentation amplifier. The tendency of a differential amplification to reject signals common to both inputs, we are calling it as common mode rejection ratio. The gain for this instrumentation amplifier can be set. We can set the amplification gain by changing this R gain in figure. The gain equation and the output equation of the instrumentation amplifier can be obtained as, this is gain equation, that is equal to 1 plus 2 R1 by R gain whole multiplied with R3 by R2. Then we have the output equation as V2 minus V1, that is nothing but difference of the two different inputs into the gain we have achieved. Now further, displacement of the EMG detecting surfaces can be done in different configurations, as I mentioned, that can be the monopolar and bipolar configuration. Monopolar configuration is implemented using only a single electrode on the skin with respect to a reference electrode. This method is used because of its simplicity, but is strictly not recommended as it detects all the electrical signals in the vicinity of the detecting surface. Next is the bipolar configuration. This basically to acquire EMG signal using two EMG detecting surfaces with the help of a reference electrode. You can see in the figure also. The signals from the two EMG surfaces are connected to a differential amplifier. The two detecting surfaces are placed only 1 to 2 cm from each other. This differential amplifier suppresses the common noise signals because that will be common to both inputs and then amplifies the difference. Now let's study what are the main characteristics of EMG signal. A raw EMG signal, it means we have not done any kind of processing. That range is 0.1 microvolt to 20 millivolt and from 2 Hz to 2 kilohertz, which may change between different kinds of people. When the muscle is relaxed, the noise-free EMG baseline can be seen. The average baseline noise must be below 3 to 5 microvolts. You can see in this figure a raw EMG recording of 3 contraction bursts of the biceps muscle. EMG spikes shape is very random, which means that each recording burst will not produce the exact shape. So you can see in this figure we have active contraction burst, then we have rest period, in between we have non-producible amplitude spikes. So these spikes are random. Now since strong superposition spike will produce when two or more motor units fire at the same time, and the location between the motor unit and the electrodes is near, then more spikes will come. Since the amplitude of the raw signal is very small, it must be amplified, and sometimes it requires more than one amplification stages. Besides that, the signal must go through to the filter in order to eliminate low or high frequency noise. After amplification and filtering, the point of interest of the signal is in the range of 0 to 10 millivolt peak to peak, while the frequency is between 0 to 500 Hz, with the dominant frequency between 50 to 150 Hz. Now next is the two steps which we are doing after getting the raw EMG signal, that is rectification and integration. Rectification is necessary. If you see this figure, a raw EMG signal, this is a raw EMG signal under no load condition. The signal does not oscillate around 0, it is somewhere near 0.2 or we can say 0.15. Offset is this is basically offset. We are calculating this offset as the average voltage, and then after adjusting the offset, the signal contains both positive and negative values that may be around 0 then. Now for that, because it is having the values in both positive as well as negative peaks, rectification involves the transformation of raw signal to a single polarity. Usually we are taking positive polarity. There can be two different types of rectification, full wave and half wave rectification. Full wave adds the signal below the baseline to the signal above the baseline, means it will add, which is negative, let's suppose negative peaks we are adding to positive. This method conserves all signal energy for analysis. And if I talk about half wave, it will delete the EMG signal below the baseline. Next step we are doing is rectification. After rectification, we have smoothing. Basically, to eliminate peaks and valleys or high frequency fluctuations, we are doing one more step in between, that is smoothing. Now next is integration. Once the signal is rectified, a method of quantifying the EMG signal is integration. It assigns a numerical value to the signal. It also permits manipulation. Basically, it forms a linear envelope procedure to measure the area under a curve for a specified period of time. You can see this example, this is raw EMG signal. It has the peaks in both directions, positive as well as negative. In this we have adjusted that offset because baseline is now at 0. Then this raw EMG is filtered. If you see, we have filtered, this is full rectification we have done, full wave, means we have added the lower peaks to the upper peaks. And then that is smooth, we have removed the valleys and spikes, and then we have calculated the total area, that is nothing but integration. Let's go through one more example of this. This is again, the first is showing the raw EMG signal. You can say that there is some baseline and we have positive as well as negative peaks, and then that baseline is adjusted and further rectified. After rectification, we have only signal at one side, we can say positive. Usually we are doing it in positive direction, and then finally we have to an integration, and this third signal is showing the integration. Now let's discuss about the EMG force relationship. An EMG signal will not necessarily reflect the total amount of force, or we can say torque, a muscle can generate, as the number of motor units recorded by electrodes will be less than the total number of motor units that are firing. If a newly recruited motor unit is close to the electrode, the relative increase in the EMG signal amplitude will be greater than the corresponding increase in force. If a motor unit is too far from the electrode, the amplitude will not change but the force will increase. That means EMG will not change but the force will increase. You can see in these graphs also, motor unit firing rate will increase as force demand increases. Initially, force rises due to increased firing rate, and EMG amplitude will also increase rapidly. As force output increases beyond the rate of newly recruited motor units, firing rate will increase. Force produced by the motor unit will now saturate. You can see it is getting saturated. But if I talk about EMG signal, that will increase more than the force output. That means they have a non-linear kind of relationships. So, this is all in this lecture. This is some references we have taken for this lecture. Thank you so much.